So now that we have all this information on one tab and we got rid of the headers, now it's pretty simple to basically put this into Partek Genomics Suite. So all we have to do is select all of this information. I select the first column here, hit Shift to the last one, and I'm hitting Control C to select all this information. Now all we do is go to our Partek Genomics Suite program, and to paste this on, all we do is go to Edit and paste to a new spreadsheet. You can save this any way you want. I'm going to save this on my desktop. Rare cancer, we'll just call this PRCC, or papillary renal cell carcinoma, and then we'll go 17 since we're looking at 17 comparisons. All right, hit save. We have missing values because some of those genes actually aren't differentially expressed in all studies, so I hit OK. And now we have our data into this program. Again, these are all the normalized Illumina scores for the full change. And again, you can see here, we have 5,000 rows. So we are looking at the top 5,000 genes in our meta-analysis based on the overall gene score. So now that we have all the metadata into Partek, what we're going to want to do is format it even further. Most of these analytical programs have a preferred way it likes to look at the data or have the data presented to it. Partek works better when all of the samples are in rows and the genes are in columns. And in this case, we have the opposite. All our genes are in our rows and all the biosets, our comparisons, are in our columns. So what we're going to want to do is reverse that. I'm also going to say that as a data scientist, this is most of what I do, is just getting the data to a point where I can actually look at it. So this is a pretty common task for any data scientist to do. In order to transpose this data, what we're going to want to do is go up to the Partek Genomics Suite window under Transform. We're going to click the last choice, Create Transpose Spreadsheet. I'm going to click on that. Here it's asking, do you want the genes in the column? Yes. And we want the biosets in the rows. This is going to go to another file, so we're going to hit OK. I've already done this before, so I'm going to hit yes. And now we have our file. Again, all of the genes are on the columns, and then our bioset numbers, including the overall gene score, is in the rows. What we're missing now is what we're going to want to do is we're going to want to basically attach all the experimental and study data to this so we'll actually know what we're looking at. Again, these bioset numbers really don't do much. So what we're going to do now is we're going to go back to our spreadsheet. We're going to copy our experimental data, which is on our summary tab of our Excel sheet, and basically merge it to this file. So let's go and check out our, basically go back to the Excel file. So if I do that, I can come down here to my Excel file. On the first tab is our summary. Again, this is all our information on all of these comparisons that we're using. I'm basically just going to select all these once again, hit copy, go back to our Partek, and then under Edit, I'm going to paste to a new spreadsheet. I don't have to really save this right now because I'm probably not going to use it very much. So I'm just going to hit Cancel, and it's going to generate it. Contains 4 missing points. Yep. And now we have all our experimental data on this in Partek. Now what we're going to want to do is merge this to our original, our transposed spreadsheet so that we can do the analysis and we can actually visualize the data. Now to merge these files, what we need to do is go up. We want this to be our destination. We are basically going to take the sample information, and I want to put it right after the bioset number in our transposed spreadsheet. So all we do is go up to File, and we go to Merge Spreadsheets. Again, here are the spreadsheets we're merging. The common terms we're using. We want to both of them have bioset numbers, so that's how we want to merge these files, given here the ID columns. And then we want all of the experimental data right after our ID column in our transpose spreadsheet. So that's what we're going to do. Everything seems to be okay, and I'm just going to hit OK. Yes. All right. Now we have our data set with not only our genes, our 5,000 genes, but also all the clinical information associated with all our comparisons. So now we're in a good place. Now we can start kind of looking at the data. Except we're actually going to have to format this even more. So number one is this top is the overall gene score, and that was computed earlier. I want to get rid of that because it's not really important in the actual analysis at this point. So I'm just going to select this row, right-click, and hit Filter Exclude. What we also have are missing values. If you look here, again, not all of the genes were differentially expressed in all the studies. So what I want to do, and trust me on this, it just works better if I take the zeros, or I mean the question marks, and replace them with zeros. So we can do that pretty easy. So I just go to Edit, Find and Replace. I don't want interleukin-8. I want a question mark. Shift. And then I replace with nothing. Or actually, I'm going to replace that with all zeros. So that should all work. And again, you can see this is very familiar to Excel. And I'm just going to hit Replace All. Yes. Okay. And now our data should be exactly how we want it. So again, if we look here, all of the question marks, things that were question marks are now zeros. So now we are in a good place to basically, we have our data, we have it formatted, we filled in all the missing values. Now let's start taking a look at this. And probably the best way to do that is through visualization. And what I really like to look at whenever I get done with a data set is look at the Pca plot. So let's do that right now.